Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled AAV Vector Shedding Assay, Best Practices in Clinical Gene Therapy Method Development. My name is Sydney Perlmutter, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion is a global leader in bioanalysis and early clinical research services. Celerion offers a unique combination of bioanalytical experience, medical expertise, clinical operations experience, and scientific excellence that gives its clients the confidence to make fast, accurate decisions about their drug development path. Celerion's molecular and cellular capabilities to GLP and GCP standards allows the support of a wide range of studies from immune-modulating drugs and vaccines to vector-based gene therapies and cell-based test articles. The close connection of Celerion's clinical research unit with the bioanalytical department allows unprecedented access to the human matrix, which is vital for timely method validation of many assays. And now I'd like to introduce our speaker for today's event. Johannes Stanta works at Celerion as the Global Director of Molecular and Cellular Biology, being in charge of the Lincoln, Nebraska and Zurich, Switzerland MCB laboratories at Celerion. In this role, Johannes strengthens Celerion's service offering with GLP and GCP compliant molecular and cell biology testing to support the development of new modality therapies such as cell and gene therapies. He previously worked in senior scientific leadership roles in bioanalysis and clinical laboratory at LabCorp and Hammersmith Medicine's research. He has hands-on industry experience in small and large molecule bioanalytical assay development, validation, and sample analysis, supporting clinical and non-clinical programs, and was leading the European Bioanalysis Forum Cell and Gene Therapy Interest Group, where bioanalytical labs across Europe share their bioanalytical challenges and best practices. And now, without further ado, I'd like to hand the presentation over to our speaker. So Johannes, you may begin when ready.
Great, Sydney. Thank you so much for the kind introduction and for having me here today. And thanks everyone for dialing in. Yeah, I'm so pleased to be here. I'm currently in Orlando at the WRAB. If anyone is also attending the conference this week, come and stop by at our booth 26, and maybe we can continue discussion about this topic about shedding in particular. Let me see if you can see my actual presentation mode. Okay, I hope that works now. So yeah, the reason really for putting this together is for many years I've had I really felt that shedding for very many clinical cell therapy, gene therapy products is more like an afterthought rather than its own a method with its own challenges being addressed separately. So I've put this together. Hopefully, it'll be very useful for many of you to really see how you could approach this assay. Also, what to ask for if you use a CRO to develop it or develop it in house. Or what's the best practice really to go about and to share it, because none of it really has been published or written really. So we at Celerion put together some methods that we can show, that we can share with you, talk to you, and talk you through the process of how to go about it. Now, just a bit of background about why we're doing it in the first place. So for the EMA and the FDA have put into their guidelines for the industry for developing gene therapy products and also other oncolytic products, that shedding assessment is a requirement really for your for your clinical development program, and also it put the definition in on what it actually means. So the shedding of a gene therapy product, it happens from patients through one or all of the following ways. So it's usually excreta and secreta, so urine, saliva, and various other fluids that you can think of as well. For example, the skin, if it's a therapy that addresses those particular wounds, and there's anything secreting from there, if there's sauce or anything like that. Now, shedding, and this is sort of why I feel sort of so strongly about this topic, is distinctly different from biodistribution, which really describes how the product is shared within the patient's body or an animal's body, and thereby the context of use and the requirements that are asked for a method are distinctly different. The main purpose for shedding as well is that it raises the possibility of transmission to untreated individuals, which is the close contacts of the patients, or also healthcare professionals who helped during the administration of the clinical trial. The FDA also provides some guidance on what the shedding assay should look like. So it should demonstrate that it's specific, sensitive, reproducible, and accurate. The FDA recommends that testing of clinical samples in a shedding assay is done in replicates to determine reproducibility, and I'll get to that particular topic a bit later on what that means practically. And the sensitivity, so limit of detection, and the limit of quantification are the key challenge really, for this particular assay. And so also a very important paragraph that's written in there: the agency does not expect the shedding assay to be validated. The assay should be qualified to meet the minimal performance capabilities and be suitable for the intended purpose. But that sort of statement they've put in there, and I agree with, but also find a bit problematic, especially for a clinical assay, where in particular the GCP really asks for and requires that all assays are validated, and particularly that you demonstrate that these are fit for purpose. And again, you need to have a fit for purpose validation approach for this to address this. But this is the text as it's written in the recommendation by the FDA. It was done in 2015, and maybe an updated version could come out. EMA, very similar. July 2009 was this one posted, but also very similar sort of approach. It's a quantitative qualified assay, so again it doesn't ask for any validation of this assay in particular, and PCR or infectivity assays are the two assays that are used or can be used, but PCR assay, qPCR in particular, assays are recommended because of the sensitivity, reproducibility, and because they're fast. Again, none of those, here specifically qPCR is mentioned, and none of these address or mention anything about digital PCR. So again, these regulations I think are still pretty good in spirit, but probably due an update. But this is the framework that we have. This is the guidance that we work towards, and sort of set the scene about how we do it, and also what would be the expectation for developing such an assay. Now, matrices as well are mentioned on what they should be or can be. So urine, feces, saliva, and semen are the main matrices. Blood, it says, is typically not typically analyzed for shedding. That's what it says in the guidance. However, it should be collected as part of pharmacokinetic analysis and also product dissemination for gene therapies that are not maybe administered into the perforation, but intramural or intramuscular or retinal administration. So you can see if the virus does go out or exit those particular tissues or areas. Now, when it comes to qualification versus validation, so as I've already mentioned, I find that sort of statement a bit problematic, and it's, I think, probably from the discussions that we've had in the industry in general, probably a bit outdated. Qualification is less and less used these days, and you sort of talk about validation, but more like fit for purpose validation. But it's still a validation nonetheless. The approach we take for this to call a method validated is to follow the sort of the three steps, the three principles of a validation, which is to plan the validation in a written plan, and where we also very clearly define and set the acceptance criteria that the method needs to meet. We then execute the analytical or experimental process of this plan, compare the results against the acceptance criteria that we've generated, and also we document all the experiments in the fashion similar to good documentation practice. And then the results are also documented, all calculations, interpretations are documented and then summarized in the final report. And also the raw data, of course, is available, and all the instruments that are being used for the validation are suitably qualified and traceable, including all the materials that have been used for the validation. So again, these sort of aspects apply, and sort of the minimum standards really I would want to see for any method that's used in a clinical trial. A good starting point really for approaching molecular methods is the CLSI guidelines, MMO6/2, which is not written for a bioanalytical method or shedding in particular or anything like that, but it gives you a very good starting point on how to approach it, what aspects do you need to consider, how to go about it, and also how to plan your validation experiments in a suitable way. So we very often refer to this guideline and references to justify the approach that we are taking. The process for a shedding assay in particular, we can split up in three distinct phases. The first phase is the design and the probe design stage. We usually go for a TACMAN, a primer probe design, and we design between 3 and 5 different primer probe sets, and all of them then need to be tested, and the best one is moved forward. Then for the actual molecular method, the qPCR or digital PCR method validation, and then once we're happy with that method that we have for the PCR, we then move into the extraction validation, which also forms a key part of the method and cannot be separated from the method in any way, and thereby these distinct phases we go through, and then after we're happy at the end, also the validation is in line with our expectations, then we consider the method ready to go into validation. And that brings me to the first poll question. So Sydney will take over in a moment, but I want to ask the audience really, which assay platform do you prefer for molecular methods? Is it digital droplet PCR or ddPCR, or any digital platform, choice P, or C, qPCR, or D, no preference, any platform as long as the method is fit for purpose? So yeah, it'd be good to see what your responses are.
All right, I'll give our audience another couple of seconds to get in their votes. And I see that a majority of our audience has voted, so thank you very much. I'm going to close the poll and then share those results with you all. So we have 28% for ddPCR, 0% for any digital PCR platform, 28% for qPCR, and then 44% for no preference. So back to you, Johannes.
Oh, brilliant! Very good. What a fast readout. Very good to hear. Well, let me just get the presentation back. That's really good to hear. So many. So again, we've got a split between ddPCR and qPCR in terms of preference here, and then the majority no preference. And I think that's exactly where I'd be coming from as well. The method has to be fit for purpose and suitable for the intended purpose. For a particular purpose. So especially with regards to shedding, I think qPCR. Maybe some people say it's outdated and prefer ddPCR. However, I think as long as the method is good and has demonstrated to be fine, yeah, totally agree. Great to hear that that I'm not the only one thinking of that. So now that brings me to the next part of this presentation, which is to really show you a method development case study. We've chosen AAV8, and we developed a shedding assay on qPCR in blood, plasma, urine, saliva, semen, feces, as you would for any a new study or viral study that you would get into your lab. So as I've said, sort of the first stage for us is the primer and probe design. And the reason why this is sort of, I want to take this sort of a distinctly different phase as well, because a lot of this is done on the computer. It can be done obviously by the client who might already have primer and probe sets tested and ready to go, but also we like to design them in-house or also sometimes optimize them. And also a key aspect here, we want to design not just one primer and probe set that we want to be really happy with, but really at least 3, usually 5. So the way we've gone about this, we bought AAV8 reference material, which is commercially available. So this one has a GFP as the expression gene incorporated here. It's a good example to sort of test it and to really show and go how to go about it. Now, the sequence obviously available, and to start designing your primer and probe set, you want to be focusing on areas that are absolutely unique to the plasmid sequence. And this will be areas where you've got an overlap, or where the expression starts, or where your promoter region is, and not an area which is, for example, purely in this area, or just purely the expressed protein. However, because you might get real interferences with the endogenous protein available, and it might not be as specific as you hope it to be. And again, so you will be focusing on the very small region where you think you'll have a suitable sequence region. So again, it's important to narrow it down, to focus on it, and to make sure as well, once you have designed primers and probes, that there's many tools out there you can use online, or also your PCR software might have a module in there to do that. But then your the short list of primers and probes really need to be tested and checked again for the specificity in the human genome. And the NCBI nuclear blast is very good for that. And also you want to check the primer as well with the NCBI primer blast. They're openly available, free to use software packages, and also the oligomer formation and secondary structures is also a very important way to look for the suitability of your primers and probes, which will then rule out and you'll get a good short list of very good and suitable primers and probes that you can try and test. Primers and probes are relatively easy to order, to receive in very good standards and quality. So again, there's no reason why you shouldn't add a big list of those, and they're relatively cheap and delivered very fast as well. So that's the first stage. You can do that as soon as you start the project, even before you've got any lab time booked in there, because that process will take a while, and you need to be ready before you start that in the lab. The second aspect, so once you've got all your primers received and the probes, you need to screen for the most suitable master mix that you get available. In our project, we chose 4 of master mixes to screen against all the primers and probes, and we go on this one here. What we really look out for here is for the lowest CT values, the highest fluorescent signal, and then also the lowest background available with any of the standards or the blanks. And I put some examples here for really what's good to look out for. So you shouldn't just look at the results that you get, the reproducibility, or just purely the CT values. You really need to look at the amplification plots as well. And it's these sort of background signals that are really worth looking out for. So you get maybe a slow creep up of some background here. This is just PBS or water, and again here as well, you've got sort of water that's creeping up. Now the software would usually correct for it, and that's why sort of the pure values might not always just pick it up. However, this is an aspect where when you move into the samples and when you start going into your limit of detection assessment here, this could really create a problem afterwards, and create you false positive results. The key really for any shedding assay is to get the limit of detection as low as possible, and really push that limit, and thereby the background is very, very important to get into check and to get it as low as possible. So for our assay, we had 2 primer and probe pairs that worked really, really well, with no minimal backgrounds really, and the master mix that was the best was the SSO Advanced Universal, and from Biorad, which showed us incredibly a clean and low background to use and move forward with very good CT values and a very good fluorescent signal as well. So we moved forward with that. The next aspect that you really want to address very, very quickly is the limit of detection, which is, as I already mentioned, the key readout for this. The standard experiment to do that is to spike your linearized plasmid at various concentrations into genomic DNA, a matrix, and then you run 8 replicates, and then you check how many of them remain positive, so we can clearly detect your signal in there, which then allows you to calculate the proportion of positive results that you've detected and clearly distinguish it from your background. You can use probit as well, but if you use the proportion, which is what I've used here in this signal here, you draw a line or you calculate the line for the 95% interval. This point here is 1, corresponds to 1, so 100%. So all 8 replicates at 20 copies have been detected in 8 replicates, and then when it comes to 10 replicates, it's only 7. So you're already dropping down here below the line, and then as you go for 6, 5, 4, 2, and 1, it sort of drops down, and you take less and less positive copies. And this intercept here you can calculate. Or you can also, it's perfectly acceptable to just draw it on paper and get that intercept done. There's various different options available, or you can calculate it, of course. And this then gives you your LOD with a 95% confidence value that you've detected positive samples. In our case, we had an LOD of 15 copies, which was okay. Probably not the best I've seen. The lowest you could possibly get would be 3, but 15 is definitely acceptable, and as shown, very good method that we were happy with to move forward. which brings us to the next set of experiments. You want to move on to sort of tighten up your method and move into the next phase, which obviously for us, precision and accuracy. For a quantitative method obviously needs to be defined. We use QC samples for this, so they are spiked prepared samples and genomic DNA on the ULOQ. Then you've got high QC, medium QC, low QC. For the LLOQ QC, because we don't want to just demonstrate it for one level, we chose 3 levels to go for: 20, 35, and 50 copies to be included, and then calculated over 6 runs the precision and the accuracy, how the back calculation of the bias is, and thereby we can establish with our acceptance criteria which one is the most suitable LLOQ. And the LLOQ by definition is the lowest concentration you can determine with an appropriate accuracy and precision. So again here, something where I feel that needs to be done experimentally rather than just theoretically. I know a lot of labs would just choose 50 as the LLOQ because many years ago in one of the regulatory documents, 50 was used as the limit of quantification as an example for biodistribution studies, which then been broadly applied to absolutely every method. However, I do think this needs to be experimentally tested and shown here. So yeah, we've chosen 3 LLOQs and all of them showed absolutely acceptable level. So the LLOQ QC at 20 copies showed acceptable performance and was the lowest level that we moved forward with this assay into the next phase. Which then brings us to a very, very big chapter of shedding, which is you have very many different matrices, and all these matrices form a part of the method. So I feel very strongly about the fact that the method doesn't just start with your qPCR and anything else we sort of don't care about. It really, the extraction is just as much a key parameter, and it's not just one matrix but several of them. So blood, plasma, urine, saliva, semen, and feces would be the standard package. So we've chosen them, and we thought we want to really push this to the experimental limits, so we know we're very, very happy with the actual method chosen, and we can categorically categorize it as well on what's the best extraction method available. Therefore, we spiked up the viral particle, which would be the actual reflection of what you would expect in your shedding experiment. You're not looking after DNA or for any sort of small fragments. What you're really interested in is the viral particles. So again, this is what needs to be spiked in, will inform you about what the extraction recovery is actually like. At the same time, it's also good to spike in plasmid and your linearized plasmid to see what are the differences and can you generate the same results overall between viral particle, plasmid, and linearized plasmid to work out, what will be if the results make sense. The acceptance criteria, I guess for many of you, you're not familiar with shedding assays or extraction of DNA, will find these values incredibly disappointing. We'd be happy with more than 50% blood plasma recovery, urine 20%, which is a sort of a known challenge with urine, and then saliva, semen, and feces as well. Over 50% recovery is considered acceptable to move forward. So you've got a method. When it comes to the actual extraction, so I mentioned earlier the FDA document actually mentions replicate testing, and I've put 2 scenarios out here which I want to put to the audience again. So get your poll clicking fingers ready. So you've got one sample here that you start off with, and then you extract it 3 times, and then run each extract once in your PCR reaction. That's option A. Option B would be you take your sample, you extract it once, and then you run it 3 times on your PCR. Or so that will give you 3 results. We then have another option as well. So you've got 3 extractions followed by 3 PCR tests. Each will give you 9 results per sample. Or option D would be all options are equally acceptable, and there's no real requirement. They're all, pretty much they're all replicates, so they're all suitable. So it would be great to hear your thoughts. A, 1 extraction followed by 3 PCR. B, 3 extractions followed by 1 PCR. C, 3 extractions followed by 3 PCR, or D, all options. Good to hear your thoughts.
Thank you, Johannes. I'm just going to give our audience a couple more seconds to get in their votes. And this is the final poll question. All right, looks like most of our audience has voted. Thank you again for participating in these polls. I'm going to share the results. So we have 22% for 1 extraction followed by 3 PCR tests, 33% for 3 extractions followed by 1 PCR test, 39% for 3 extractions followed by 3 PCR tests, and then 6% for all options are acceptable.
Excellent. Wow, that's great to hear. So I think I need to go back to the screen. Here we go. Interesting. 22% option A, which I have to say is my least favorite option. Extracting once, for me, that's a singlet for me. That's not the true replicate. And then 3 times on the PCR. So we've seen here as well on the precision assessment here, the precisions of the replicates are actually really nice and very tight, in the region of 20%. So I don't feel we need to sort of prove that again with every experiment. So A would be my preferred option. It's a balance between how much effort you spend on the method and also the PCR, so what your throughput of the sample is going to be in the end. But yeah, 39% think so. 3 extractions followed by then 3 replicates. So yeah, the throughput per plate per sample is then obviously massively decreased. But yeah, good to hear. Great to know. I'm glad I get your input, your thoughts on here. It really helps shaping the experiment and also knowing about the expectations generally out there. So for our methods, we thought, well, we test all kits that we can sort of get our hands on the market, and there's plenty of them available. And they all have in their kit inserts various different specifications on what they allow, what they claim the extraction kit can be used for. So QIAGEN blood and tissue kit can be used for blood, plasma, saliva, and semen. There's only 1 stool kit from QIAGEN, but then Asay and Promega have a stool kit as well for feces that can be used. Urine as well, there were 3 kits. For plasma, there's 4 kits available, and we thought, well, we put them all to the test. We'll test blood against all the kits, and we did that in 3 individual separate extraction experiments, where we all tested them against each other with the same spike solution, and to work out which will come on top, because they all claim to be good and suitable. And the results were very surprising for us, and really showed the importance of this particular experiment, which again, for any future development study we would repeat, and we can't take those results for granted. So on top came the QIAGEN blood and tissue kit for blood, plasma, saliva, and semen, with a percent recovery close to 100%, semen slightly less. It does take a slightly modified protocol as well, where you need to add a DTT. The limit of detection, so, and this is sort of very important, and this is why this extraction kit needs to be part of the development and the validation process as well, because the limit of detection changes with every kit. It's not the same for 2 kits for 2 manufacturers, because the amount of matrix you can add per kit varies dramatically. So the limit of detection, which is here expressed as genomic copy per microliter, is different for each and every one of those methods, and this is the actual LOD. The PCR limit of detection is really only the technical aspect for your PCR reaction, and that's 15 copies. But it's 15 copies, and you need, okay, if you want the concentration, 15 copies, well, how much was the input that you've had? So here, the genomic copies per microliter between 1.3, 2.7, 1.6, and semen 4.5 genomic copies per microliter. You can and that really creates a very important aspect for describing and defining your method. So it needs to be repeated for every product, for every plasmid, and also for every validation and for every prime and probe set that you use. For feces as well, this was a very interesting one. We knew the recovery was always very high, so we weren't too worried about that. And that makes sort of sense from a principle point of view. Obviously many people will know that the inhibition can be enormous for feces. However, the extraction kits are now so good as well that really the recovery was very good. 7.3 genomic copies per milligram of matrix, because this comes as a solid. Interestingly, so the Zymo kit and the Promega kit both claim to be suitable for feces, but we had absolutely no recovery from those. They were below the limit of detection that we could find in the PCR. So yeah, I'm very glad we tested that and showed that this was not suitable for our particular product. Urine as well, as I said, so we're happy with a 20% recovery, and we had 2 candidates that we thought would be suitable. The Zymo research kit with a 30% recovery, and then also the QIAGEN viral RNA mini kit as well, 37% recovery. However, the input is very, very different. On there you can add in there, and urine you're not limited really by the volume, so how to get most of it out makes a big impact. And 1.1 genomic copies per microliter for urine, even though we only have 30% recovery, we felt was the one to go for, and was the winning kit to use. The Zymo Research Quick DNA Mini Prep Plus kits as well, they were also suitable, so it showed good recovery, consistent recovery as well. You're allowed to load a lot more onto those kits, so that's why the actually LOD is lower compared to the QIAGEN blood kit again. So this could be an aspect for deciding on which one you want to go for, or if you want to stick with one kit and have that used throughout. Again, that could be a justification for going for one kit over the other. But until you've actually processed them all against each other and trying to test them, it's very difficult to choose. And the kit inserts really have to be tested experimentally to allow you to actually get to the truth of your limit of detection. Once you've established that, you can go into the assay validation. We've set up a streamlined validation workflow, which comes in 6 distinct steps, and 1 step needs to be completed before you can move into the next step. It starts off with just looking at the qPCR calibration curve, limit of quantification, linearity, and the range, with acceptance criteria, which are sort of pretty much standard in the industry. Specificity as well, you want to see no amplification in presence of human genomic DNA, showing that there's no cross-reactivity of the assay. And then your LOD experiment, very, very important again. So I'm sure you can use the same approach. Again, your 8 replicates, and you detect at which point you can still detect with 95% confidence a positive result. Collinearity, you want to make sure your genomic DNA is collinear with your linearized plasmid, and it overlaps, so you've got confidence that your calibration curve, which is done with the linearized plasmid, is a true reflection of your genomic DNA that you've got available. And then 3 accuracy and precision runs using QC samples, similar to what I've shown earlier. Obviously you could use more than that. This is just a minimum, so could use 6 as well. The CV and bias, we usually set at 35% and 45% of the LLOQ, and then once you've got that nailed down, you can then move into extraction experiments. The recovery here, I'd say between 50 and 200% is something you want to aim for. and then the CV and bias. Here you need to calculate it as the total error. So you've got your PCR error and your extraction error, and my limit for that will be 45% for the total error to be calculated. And then, yeah, once you've gone through these experiments, they're very similar at this stage of the validation for each and every product. So you can run through them very fast, and you'll make the right decisions if you stick to the acceptance criteria. Once you've got your method nailed down, actually think about the sample. Well, you probably think about that beforehand already, but like once you think about reporting of your results, you'll also find that shedding assays have a unique requirement, really, in terms of the analysis, and that's distinctly different to most other assays that we run in a bioanalytical lab. So the guidance actually talks about how the reports, how the results should be reported, and when it happens, and also it gives you even a sampling schedule, and this is really what most people will put into the protocols. So again, if you support the clinical protocol, if you write the clinical protocol, this will be the expectation, and most clinical protocols for shedding gene therapy will look like that. So you start on day 1, day 3, day 7, day 10, and then after that you've got weekly testing. Very importantly for this particular assay is the analysis of the samples should continue until 3 consecutive data points are below the limit of detection. So here we get into an area where the turnaround of your samples is hugely important. Those samples, especially when it comes to the 3 limit of detection, below the limit of detection, negative results, when the samples come in, you do need to have to provide a result very, very fast. So because you need to inform the clinic if the following week another sample should be taken, or if they can come off the contraception. And so it's not straightforward. You're not going to just get all your samples at once every 6 months, and then you run them and you report the samples into a database. This has got unique requirements. So this has to be really carefully thought about when you set up your reporting structure and your sample analysis plan to be able to deliver that. And what would that sort of generally look like? Well, these are the results, again, that I've sort of made up, but that you can probably expect for most AAV clinical trials. So your day -1 will be below the LOD, so it's negative, of course. Then the dosing starts, and then your results will start getting up. Now, most of our methods go up. The range is enormous, to the millions. However, you don't really need that much for a shedding assay. The results are usually not that high. So again, that also focuses on where you need to focus your range when you validate it. You don't need to be super accurate in 10 to the 6. You'll never see samples in that region. But the lower level, LLOQ level, up to a few thousand, this is where your samples are going to come in. Samples are unlikely to really sort of drop significantly in any of the matrices between week 1, 10, and 11. Here's an example. I chose a week 12. But it could be week 20. You start to see a drop below the limit of quantification, and then weekly after that below the limit of detection, and then you need to be focused and on it with your sample analysis, because these 3 consecutive below LOD results make a huge impact. So you want to be timely in reporting that. You don't want to continue too much, and you don't want to not be ready when you get those samples. But planning of this is not too difficult, because when dosing happened. So in this particular area, I'd say between, I don't know, week 10 to 30, this is where the turnaround time for those assays needs to be super fast and quick, and you need to look out for those. Again, once you're prepared for it, once you plan for it, it really is not a big deal. If you're not familiar with the unique requirements of a shedding assay, this can catch you out possibly. So what's the conclusion? Shedding assay have a unique context of use, and I hope I've sort of explained that here to you. So inhibition from different matrices is possible, so that's something you really need to look out for. Different kits need to be evaluated and validated as part of your method. Don't just trust the kit insert and think all the validation will be good. Fast turnaround is needed to inform the next sample collection from trial participants. While I'm saying that, and while this is obviously hugely important, I do not think that this is part of an IVD or a clear assay for various reasons, and I'm happy to discuss that further. The PCR validation should focus on the limit of detection. That is true for a qPCR as well as a digital PCR or digital droplet PCR. This is sort of the key focus on it and the lower level of the range. And yeah, the extraction kits need to be tested, and also the reproducibility and also calculated in terms of the amount recovered per milliliter or milligram of the matrix that you work with. And that pretty much brings me to the end. Acknowledgement. Sylvia and Andrea worked very hard on this in our Zurich facility and generated all the data in the lab for this, as well as Petya, also big support in our Zurich facility, and then Petra, our executive director, who allowed us to pursue this project to show and share this information with you. So big thanks. I want to hear your questions and the discussions, and I look forward to your thoughts.
All right. Well, thank you very much for that insightful presentation. Now, I'd like to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of the webinar. So I've already got some questions, and we'll begin with those. So our first question, how long does it take to develop and validate a shedding assay?
Very good question. We've got a very streamlined process set up. So for a lot of the experiments, we use design experiments which are already templated, and we know how to approach them. We just need to plug in the right primers, the right master mixes, and then just follow the process experimentally through. So yeah, this can be done really in a matter of weeks. Same with the extraction protocols for the kits. Most of the kits we've got available, we've got in stock. We can really start moving on this very, very fast. And then validation as well. I'd say a matter of weeks. This can be completed. There's very little points in between where we need to pause and sort of take a step back and think about what we're going to do next. Because of the unique context of use of shedding assay, this can be, very easily pre-planned and put together, and then just processed and done in the lab very, very quickly.
All right, thank you very much for that answer. Another audience member would like to know, in LBA and LC-MS or MS assays, LOD is not utilized. What value does it provide when the LLOQ is what is important for reporting the number of copies or concentration?
Yeah, well, great question, and I totally agree. So the context of use of LBA or LC-MS method is different. It is really to provide quantitative results in the region of 15% or 20% for LBA to inform usually in bioanalysis about the PK assessment. So that's the main aim. The context of use for the shedding assay is really, we're doing a quantitative assay, but it's the qualitative result that has the main impact on the clinical trial. So for how long did you have to follow the shedding in the matrix before it went below the limit of detection? The results that you get in the first few weeks for the limit of above the limit of quantification give you a loose information about how successful your product was in circulation, but really I've seen very many agencies ask for it or comment on it, and I think it's a nice to have rather than something that you really will push on from a quantitative point of view. So the key for it, and if you're in the lab analyzing those results, you got the limit of detection is really where most of your results are going to be, and you're going to be counting: is it 2 in a row? Is it 3 in a row? Is it just one LOD? And if it's one below the LOD, which is a negative sample, you're going to have to plan very, very carefully when the next samples are going to come in and when you have to turn them around. So that is where the focus, that's where the results are going to be. That's what everyone is going to be interested in from the sponsor point of view and from the lab operations point of view. And that's why this criteria for this particular assay, for this context of use, for me is the most important parameter.
Thank you for that answer. Another audience member would like to know, what would be the reason for shedding result to revert back to a positive result after a 2 times negative?
Yeah, very good question. And I've seen this and the frustration that comes with it. Yeah, what are the reasons? So I think there's a lot of hypotheses out there, a lot of theories out there. A lot of things have been discussed and thrown about in very many project meetings that I've been involved in. My personal opinion is that a lot of it is assay driven, is assay dependent, especially when the method and the lab hasn't carefully validated and chosen the extraction a method, which what I found has been the biggest frustration with a lot of the validated assays that I've found, where just a kit is used and then processed through. And I think the extraction as well can be different from one week to the next, from one day to the next. And I think that could then obviously influences the recovery, and that then influences if the assay if the result can still be picked up within the PCR reaction. And one sort of result above the limit of detection will make it automatically a positive result again, above the limit of detection. So again, there's sort of an influence of that aspect where the sample collection together with extraction and then the highly sensitive qPCR method that you have in the end contribute to that sort of uncertainty within that result. So I don't believe the results are genuine. So someone will not be not shedding and then shedding again and then not shedding again. I think the issue here is definitely, from my point of view, technical, experimentally, and based on the extraction limitations and the sensitivity of the methods.
Thank you. Another audience member would like to know, do you run the QC panel in buffer genomic DNA or carrier DNA?
Very good question. Yes, and we thought about this very intensively, and we chose genomic DNA as our standard matrix to be used for various reasons. The main reason, my justification, is that is the most suitable for running in the qPCR, and it's also relatively easily available and producible and reproducible, and people can understand it and get their head around it. And obviously it will show you by selectivity as well immediately, because you have the genomic DNA there, you've got it present, and for me that makes the best surrogate matrix to be used.
Thank you. Our next question, are short-term stability and long-term stability assessments be performed during validation?
Yes, very good question. And I haven't put that on the slides, but the answer is yes. It has to be, it should be included as well, of course, to cover the duration of the storage. It's not too difficult to be done as well. Also, most samples will be analyzed fairly quickly, so you don't need to establish years and years and years of frozen stability. But it does need to be assessed. It should be assessed, and usually a 3-month stability assessment, which fairly quickly done, should really cover all the sample analysis that's needed for the duration. We've also found, I thought I want to add to that, during the stability assessment as well, and the freeze-thaw assessments in particular, that you can see differences potentially of the recovery after you've done the freeze-thaw. but they're not genuine, they're artifacts, and a lot of them we found were due with how you spike your reference material into it and what matrix you use to spike into this, rather than a genuine loss of stability of your genomic DNA from your viral genomic DNA, which, I mean, we cover it in validation. However, it's very well known that the stability of these samples, is there, should be given, and shouldn't be a concern. So if you run into stability problems or freeze thaw problems, it's probably to do with the experiment rather than a genuine problem.
All right, we have time for one more question, and that is there a minimal volume of sample you require to perform these assays?
The answer is no. There is no minimum volume. I think it has to be, sort of balanced in terms of also it will have a direct impact of your limit of detection as well, of course. I would say, a standard collection for most of these matrices is not too difficult to get. Obviously, urine, you've got loads. Blood collection as well, I think the minimum will be, we'd say 150 microliters, which again is usually very, very easily obtained. Saliva, usually not too difficult, easy as well. And yeah, also with feces as well, we don't need loads. Really, a small quantity will really allow us to go and do the extraction. And yeah, most extraction kits that we use are somewhere in the region between 100 and 500 either milligrams or microliters that you can do. I mean, well, so there was one kit that we assessed where you can actually use 40 milliliters of urine, and you can add some beads in there, which will take out and extract the DNA for you, which you can centrifuge, and then you reconstitute it, release the DNA, and thereby you have the potential of actually incredibly concentrating the DNA that's present within the urine. And we were initially quite excited about this product. We thought, oh, this could be great. This could be a game changer. However, your analyte is not present in free DNA swimming around in urine. Obviously, you deal with the vector particle, so the recovery and percent recovery and also the overall LOD improvement from this product was actually incredibly disappointing.
All right. Well, thank you very much for those answers. We've reached the end of the question and answer portion of this webinar. If we couldn't attend to your questions, the team at Celerion may follow up with you, or if you have further questions, you can direct them to the email address on your screen. Thank you, everyone, for participating in today's webinar. You'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event. And a survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now I'm about to send you a link in the chat box, and you'll be able to view the recording of this event on this page, and you can share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join us in thanking our speaker, Johannes Stanta. We hope you found this webinar informative. Have a great day, everyone.
Thank you so much, everyone.